All right, folks. So I'm going to post a second um, lecture on lab-related material, and so this is on one of the more significant of the second half of the lab, which is what we call IMViC testing. Okay, so um, let's just get into that, and I'll pull this PowerPoint up and we'll look at it. Go. And this, again, by the way, is on eLearn as well, in the lab shell for the course. If I remember, I'll put a link to it up on the lecture eLearn shell so that those of you who would like that can have it. But IMViC testing. Now, in the previous lecture on differential and selective media, I talked about EMB and McConkey augers and how they are mainly used to select and differentiate gram-negative organisms, specifically those that live in the gut, forms that are often referred to as coliforms. All right. Coliforms. There are other names that are given them, and each of those names really they don't always mean exactly the same thing, like in Enterobacteria, ACE, and so forth. But coli in this name, C-O-L-I, that's referring to the colon. So these are organisms that are most commonly found in the gut. A few of them, it's been found, can occur elsewhere. So it's a little bit of a stretch to say all of them are in the gut. But anyway, you need some means to be able to detect the presence of these, because it could tell us whether food is contaminated or water is contaminated. And that obviously could lead to some illness if people were to ingest those contaminated foods and water. So let's look at some of these additional means beyond using the McConkey and EMB auger to look at these gram-negative organisms, these coliforms, and learn more about them to differentiate them further. So we have these IMViC tests, and you can see that's an acronym, obviously. Each of those letters stands for a different thing. And so here they are. So the I stands for indole testing. We'll talk about what indole is here in a moment. Some organisms can produce indole and others can't. I'll just tell you right now, indole is used as a communication substance. In the lecture on chapter 7, where it talked about biofilms and quorum sensing and the production of inducer substances, indole may be one of those. So it's used in communication between, we think, individual organisms in a biofilm or in a colony. And some organisms can do it and others can't. The testing that we do for indole, by the way, involves use of a particular medium that not only lets us test for indole production, but other things as well. So the results of this IMViC series of tests are not limited to just these 4 things. There's more to it than what is shown here. Usually those are the ones that are highlighted. And then we've got what's called a methyl red test. This is a test that looks at metabolism of sugars. We'll see what that's about. And likewise, we have a similar test which also looks at metabolism. It's sort of always given or always done in concert with methyl red testing. They go hand in hand. Organisms that are positive for the methyl red test typically are negative for Voges-Proskauer and vice versa. So we'll talk about what the Voges-Proskauer test is looking for. And then the C in IMViC stands for citrate utilization. Some other things that we get, I'll mention, with indole testing, we can also see whether the organism uses sulfur specifically whether the organism is a sulfur reducer, sulfur reduction. Test for that, and we also test for motility, which is the organism's ability to move, which would be indicating that it's flagellated. So does the organism have flagella or not? That's another thing we can learn as part of that indole testing. Okay, so we'll talk about indole testing first. So indole is a byproduct of the metabolism of this stuff here. So tryptophan is an amino acid, right? It's very abundant in foods that contain proteins and peptides. Turns out that there are some microbes, some bacteria that can utilize tryptophan amino acid. They take it up, and if they have a particular enzyme known as tryptophanase, they will deaminate the amino acid, which literally results in the removal of this section here. You can see contains an amine group or amino group. That's all removed, and this is the residue that's left behind. That's the indole. And so it's a two-membered ring, one ring with 5 corners, another with 6 corners. You get this little nitrogen-containing side group there. That's indole, and it's not used as a substance that provides energy or carbon, but is, as I said, utilized as a communication substance. What the organism does with this part up here, I'm not sure, but this I know is for communication. So we have a way to test for the presence of that stuff. We add a reagent to a tube containing organisms growing in sim agar. We're using the sim medium, which is an agar. We use a reagent known as Kovacs. And I'm remiss, I don't remember the exact chemical composition of Kovacs reagent, but in the presence of Kovacs, this indole will give a cherry red color. Right? So that's one of the things we can find using sim agar is whether there's indole that's present. In addition to that, there's this sulfur reduction that I mentioned. Now the sim agar, it's an agar that is made with a smaller percentage of medium suspended in water than we would normally use. So it's not going to be a real solid agar. It's not something you would pour into a plate and try to streak. Couldn't do it. Instead, it's more of the consistency of like pudding or yogurt or something. And in addition to the tryptophan that the organisms could break down into indole, there is sulfur that's included in this medium. In the form of, I think it's just elemental sulfur. There's also iron in here, small amounts of iron. And that typically just sits there. It's inert, unless organisms are reducing sulfur. So here's what happens in sulfur reduction. Here's sulfur. If it gets reduced, electrons and hydrogen ions are added to it, and it becomes H2S. So this is hydrogen sulfide. H2S, in the presence of iron, will give a black color. Black precipitate, right? Well, that's what we're looking for there, if there's sulfur reduction. That's another thing you can spot, right? So you put an organism in this, grow it up, If you see black color, boom, you know it's a sulfur reducer. If you then apply the Kovacs reagent, add it to the growth, you get a red color, boom, you know you've got indole. All right, I'll go back over all this stuff, so don't worry. And then the last thing is motility. Okay, you can see here the acronym SIM, right? Sulfur indole motility. Motility is assessed using this auger. Here's the tube of the auger, the SIM auger. When we inoculate it, we take an inoculating loop that has been de-looped. In other words, it's been straightened. There's no loop in the end of it. It's just a needle. You stab this needle straight down in there and then pull it out. And the organisms then are left behind. And if they grow, you'll see coming off of that little pore that you formed in here, you'll see growth going out into the surroundings. It's thin enough that flagellated organisms can swim out there and create some turbidity. If you don't see that, you know, if you stab the auger and then withdraw it and look for growth, and you don't see it extending out away from that, if it just stays limited to that little spot right there, then you know it's not motile, right? It doesn't have the ability to move, right? So we're testing for indole. A positive test for indole would indicate that the organism has tryptophanase, and we would see that as a red color in the presence of Kovacs. If the organism can reduce sulfur, form H2S, we'll see a black precipitate. You don't see a black precipitate, it's not a sulfur reducer. And then finally we can check for motility by stabbing the auger and looking for growth that extends out into the auger, creating turbidity out there. If you don't see that, it's not motile. So, you know, we can differentiate coliforms, especially the Salmonella and Shigella species. They're commonly studied utilizing this particular medium. Sometimes people use it not as part of the IMViC series of tests, they just use it alone and study Salmonella and Shigella that way. There's several species of each of those, and they do different things, and commonly indole testing and other things are used to investigate those. All right, so here we've got a couple of indole test results. We've got two different species. This is Salmonella. Salmonella, the S stands are for Salmonella. Salmonella typhimurium, which is the one that causes people to get Salmonella food poisoning and that sort of thing. And you can see there is a dark color in here that indicates there is H2S reduction. Okay, so H2S and iron gives us a black color. So this was made, that indicates that there was sulfur reduction. You don't see a lot of growth up here because those organisms are not going to grow well in the presence of oxygen. Also, the Kovacs reagent was added there, and so that's going to produce a little band of light color at the top. And you can see that there's no red, so there's no indole production. So we'll say indole negative for that result. It's H2S positive. It reduces sulfur. And because you see the black color throughout down here, this whole thing, it's spread all the way through, we can deduce that it's probably motile, right? So indole negative, H2S positive, and it is motile. That means it is capable of having motility. So Salmonella has that result, at least that species. And then this is another common coliform, and it is Proteus vulgaris, right? And this one has exactly the same result as Salmonella. So it has sulfur reduction. It is motile. The only difference is it does, in fact, produce indole. And you can see right here at the top, see the red color? Kovacs was added, and we've got a little bit of red color, so we're going to call that indole positive. So it's indole positive, positive for sulfur reduction, and it is motile. All three of those things apply. I don't have any other pictures of results of this kind of testing to show you, but you could imagine some other possibilities. One would be no black color in there at all. Yet, cloudiness throughout. So that would be an organism that's motile but is not a sulfur reducer. Then, the presence of red or not in the top would tell us what the indole response was. Sorry, I erased it all. But yeah, this one is indole positive. I'm sorry, I spelled that wrong before. Positive. All right. This is indole negative. Okay. All right, so you got the indole testing. As I said, you get a lot of mileage out of that because using SimAuger lets you test also for sulfur reduction and motility. Pretty cool. Now we've got the MRVP testing, and this uses a special broth. It's really not a whole heck of a lot different than nutrient broth. It contains peptone, which is a source of nitrogen. That's always going to be present in nutrient broth, any kind of broth. Dextrose is a carbon source. That's glucose, carbon and energy source. That's glucose. And then we've got a phosphate buffer, and all that does is keep the pH regulated to prevent it from shifting up and down and affecting the quality of the results. So what we do is we take a given organism, an unknown organism, one that we're trying to, more fully characterize, and we take 2 tubes of this broth, all right? So 2 tubes of MRVP. These are both MRVP broths, 1 and 2, and we're going to put the same organism into both of those, all right? And then one of those later will be tested with the methyl red test after we get some growth. And that same organism, we're going to perform the companion test, the Voges-Proskauer test. All right. You can't do both of these tests with just a single tube of organism. All right. Once you complete one of those tests, that tube is done. So the upshot is we have to have replicate tubes so we can do these separate treatments. All right, so the methyl red test is simple. Once you get some growth in some MRVP broth, it takes about a day for that to happen. You can add very simply this substance known as methyl red. Right, and this is a pH indicator. I talked about phenol red earlier, and then there was the neutral red in the Conky auger. This is a different one of those substances. This is methyl red. And methyl red is weird. This one will not turn yellow in the presence of acid. It'll actually become even more red. All right, so that's unusual. Usually we expect, I guess, a pH indicator to change color in the presence of acid, but this one doesn't. This one stays red. If it turns yellow, that means alkaline byproducts were produced, right? So red is indicating a positive test. When you add methyl red, if it stays red, it's positive. It's positive for mixed acid production. So the dextrose was broken down into acidic byproducts, what we'll say, right? So that's a positive methyl red test. That's one direction in which organisms can go. Now, if they're positive there, you almost don't even need to do this Voges-Proskauer test because the chances are extremely good that the organism is going to be negative for that. But we always do it anyway. We always want to do it because it's possible you could have an organism positive for both of these things. Not likely, but possible. Okay, so the Voges-Proskauer test tests for a different outcome, right? So dextrose, let me just go through what happens with dextrose. Here, let me pull up another. Let me pull up another slide, and I'll just use that whole slide. All right, so here's what happens. Let me use a different color. Get tired of that one. So dextrose is actually glucose, okay? It's just an optical isomer of that. And what all organisms do with glucose is they pass it through a series of 10 reaction steps in the cytoplasm of the cell, and separate enzymes there that take this dextrose and break it down in a series of steps, so it's gradual, and they make a little bit of ATP along the way, and they make a little bit of NADH along the way. And then what happens with the dextrose is it's split into two pieces that each have 3 carbons in them. And this is called pyruvic acid or pyruvate. So dextrose has 6 carbons. Pyruvate has 3 carbons. And the pyruvate is still good stuff. It still has some utility. It can be broken down further. So what mixed acid producers do is they run this through a series of reaction steps, and they make, more ATP, and they make more NADH, and they make FADH and other stuff. We don't need to worry about that. But the end result then is the production of a mixture of acids. Now, what other organisms do that aren't, I apologize for that mess, it's awful. So that's one route, all right? That's one possible outcome. Other organisms take that pyruvate and they run it through a different metabolic pathway that results in the formation of a substance known as acetone. And there's actually things beyond that, but there's a series of steps beyond that. But this acetone in this pathway, by the way, this stuff's called butylene glycol. I've forgotten the name temporarily. So, butylene glycol. And this is called the butylene glycol pathway. This is what the organism's trying to produce. But a way that we can know that it's producing that is to test for this stuff, acetone. We don't have a test for butylene glycol's presence. We do have one for acetone. If we see acetone, we know it's utilizing this butylene glycol pathway, if that makes sense. So these organisms that do that are different from the organisms that do this pathway to make mixed acids, all right? So we're differentiating organisms. Is it a mixed acid producer, or is it a form that utilizes butylene glycol metabolism? And the way we know it's using that type of metabolism is that it's producing acetone. Right? Hopefully that makes sense. It should. So that gives you some rationale there about what we're trying to accomplish. why it works. So the Voges-Proskauer test is testing for whether or not the organism utilizes a butylene glycol pathway, and the substance that is our marker, that's our indicator of that, is acetoin. The reagents that we add to the tube of MRVP broth to check for this are two of them, Barrett's reagents A and B, right? And one of them has sort of a mothball smell, and the other one is potassium hydroxide. So you got a little bit of potassium hydroxide in there. I can't remember what the chemical substance is that's in that. Oh, naphtha. So alpha naphtha and potassium hydroxide. That's of really no significance. Just know Barrett's reagents A and B are used for testing in this Voges-Proskauer test. And the end result, it's really tough to see this. It's a challenging test because the results are subtle. But if it's a positive test, after addition of Barrett's reagents A and B, and you have to add them in that order, by the way. A goes in first and B goes in second. Then you incubate, and if you see red going up, sort of almost like a burgundy color, almost like some red wine. You see that, it indicates it's utilizing the butylene glycol pathway. Acetoin is present. Butylene glycol pathway is what they're using. If you see a copper color, like the color of a penny that's got a little bit of a patina to it, that's been tarnished a little bit, an old penny, that sort of brownish look, then you know that it's negative for that. And it probably, if it was negative for this, if it was negative for Voges-Proskauer, it's probably positive for methyl red. All right. Here's two separate organisms. One E. coli and this organism E. cloacae. Cloaca is the combination anus and urethra. The same passage for both kinds of materials that we have in birds. And so there's some forms of bacteria that inhabit that part of the gut of, say, chickens and that sort of thing. So I think this is Enterococcus cloacae, I think is the genus that E stands for. At any rate, they're coliform organisms. And you can see here they've done the methyl red tests for those two organisms using Voges-Proskauer or MRVP broth. And the red color here indicates that this is a mixed acid producer. Right? It's positive for that. And then the yellow color here indicates that no, it's not forming mixed acids as a result of dextrose metabolism. It's likely using a different pathway. So we can go ahead and do the Voges-Proskauer test. Like I said, it's kind of redundant to do that for organisms that have already tested positive here. We do it anyway. And you can't really see this because it's so dark. Remember I said it's really sort of subtle. But that is a brown color. So that's definitely brown. There's no hint of red in that. And so E. coli is negative for that particular test. So it is not a mixed acid producer. On the other hand, it's negative for the butylene glycol pathway. That's what it's telling us. It is a mixed acid producer. It's negative for butylene glycol. Over here, if you look in the top, you can kind of see a hint of red in there. So that's red, all right? And so that tells us it was positive for this test. This is an organism that utilizes the butylene glycol pathway. See those results tie, these results sort of tie together here. All right, so there's that, the MRVP tests. And the last thing in the IMViC series of tests that we want to know is whether or not the organism utilizes citric acid. And citric acid can be another carbon source. So some organisms have the genetic means by which to produce enzymes to do that. Others don't. Others are not equipped to utilize citrate. So we can differentiate on the basis of that as well. And so we use this agar. It's called Simmons citrate. So it's a nutrient agar. It contains the substrate, citrate. And then it's got a pH indicator, right? It has ammonium phosphate, which when it's metabolized will produce products that raise the pH, elevate the pH. Okay? It goes way high above neutral. So that's what the ammonium phosphate's in there for. The pH indicator is royal blue. Now, royal blue starts off green. In a neutral pH condition, it's going to be bright, sort of, not really bright, but forest green, an obvious green. That's what I'm trying to say. Definitely green. When organisms grow on this and they metabolize ammonium phosphate and produce alkaline end products, they combine with that royal blue and they actually cause it to look royal blue, right? So this is an outstanding test. It's one that is very reliable and it works. And it does nearly every time. It's pretty striking. So here we've got a tube with Salmonella typhimurium, and you can see this blue color in here, a royal blue color. That indicates that ammonium phosphate was metabolized, the citric acid was metabolized, and it's raised the pH considerably. So this is a citrate positive organism. The agar didn't look like that to begin with. It turned blue as a result of citrate utilization. This is what it looks like to begin with. It's got the green color. And because the organism growing on that didn't change it at all, we know that it's negative for citrate. Well, here it is written down here. Negative for citrate utilization. So E. coli is negative for citrate utilization. Doesn't ferment, doesn't metabolize citric acid. So there's that. And that's basically all you can do with that test. You can make slants like these, or you can use plates of Simmons citrate agar. That's what it'll tell you. But it's a really reliable, really useful test because it differentiates E. coliforms very cleanly, very, very nicely into different categories. That, I think, maybe it. Yep, that's it. So there's the IMViC series of tests. All right, and once again, that's incredibly important in studying coliform organisms. Be able to implement those along with things like using the Conky agar and EMB agar. Gives you a lot of data upon which to make decisions about, what the organism is that you're working with. All right, so I'm going to go ahead and stop that there, and I'll have a separate little sort of quiz/exam on that and use that. And then I've got one more lecture that I'm going to do here. And if I can get my cursor to work. All right, I'm going to stop there.